Hello, I am Saylor Williams, and I am the lab manager for Dr. Angie Serrano at Boston University. And today I'm going to be presenting on supporting patient-focused research through a KAT6A and KAT6B iPSC bank. To start off, I'm just going to be talking about three main questions that are going to be themes throughout this presentation. And the first one is, what are induced pluripotent stem cells, also known as iPSCs for short? What is an iPSC bank? And how is that going to help us as researchers, patients, and families learn more about KAT6A and KAT6B? Most people are more familiar with stem cells, which have the ability to develop into other types of cells in the body. iPSCs, also known as induced pluripotent stem cells, are very similar to stem cells in the fact that they also have the ability to generate into other types of cells. However, they're different because iPSCs were taken from an individual and then reprogrammed back into this embryonic-like state where they then have the ability to differentiate into other types of cells. And this cartoon here gives a very brief overview of how this is done. You start off by collecting cells from an individual, and you go through this process called reprogramming, which is a very specific protocol where you can create an environment for the cells where they reprogram back into that embryonic-like state. And then once you have these iPSCs, you can do something similar called differentiation, where you give the cells a very specific environment, and they are going to differentiate into other types of cells in the body. This gives researchers a really unique opportunity to work with cell lines in their work that directly reflects the patient population, because you started off isolating the cells from the individual and then reprogramming them back into iPSCs, which you can then use in your work. So what does this look like in the context of an iPSC bank? So our goal is to collect as many samples from patients as possible with KAT6A and KAT6B. We understand that there is variety in the clinical manifestations for both, and that is why we want to collect as many patient or samples as possible to really reflect the patient population. And so what we're going to do is we're going to do a blood draw from these patients. And we understand that doing a blood draw can be stressful and cause some discomfort. So we're really trying to do everything we can to make it as comfortable as possible. And we're hoping that it will be less invasive as some other ways to collect a sample, such as a skin biopsy. And then once we have collected these blood samples for a lot of different patients, we are going to isolate peripheral blood mononuclear cells, which are known as PBMCs for short, and these contain the genetic information, which is going to allow us to reprogram them into iPSCs, which is that process that I had just talked about before. And once we do this for all the samples that we have, we can store them at an iPSC bank at the Center for Regenerative Medicine. So when we refer to an iPSC bank, it's kind of similar to what you think about when you think of a bank, when you store something nice and safe. So that's what we're going to do at the Center for Regenerative Medicine, is once we have all of these cell lines, we're going to store them safely in an ultra-low freezer where we can access them when we need to. And I'll talk more about the Center for Regenerative Medicine in the next slide. And our goal with this iPSC bank is that we can share these cell lines with scientists all across the world, so they will be able to use the cell lines in their work that reflect the patient population for KAT6A and KAT6B. Once they have these cell lines, they can use them in their work to differentiate into different types of cells in the body. Later on in the presentation, I'll talk about what we're doing in the Serrano Lab with differentiating them into neurons. And you can actually take this a step further and differentiate them into organoids, which are 3D structures of those cells. And then you can use those organoids and cells in your work to test your hypothesis, add some research questions, and investigate therapeutic approaches, all while doing these with cells that reflect the patient population. So as I mentioned, this is going to be held at the Center for Regenerative Medicine, also known as the CReM, and we had a show and tell there yesterday, and I know a lot of you were able to come, which was wonderful. And the CReM has a commitment to open source biology, meaning that every resource that is generated in the center is then shared with researchers across the world. So that is referring to our iPSC cell lines we develop, but also the protocols. So you can actually go on the website and it will list all the different iPSC cell lines we have, and you can go in and request them. We currently do not have any of the KAT6A or KAT6B cell lines up yet, but we're hoping to do that very soon. And we're really hoping that this helps increase communication and collaboration among scientists across the world. And this is a picture of where the cells are going to be stored in the freezer once we start doing them. So if you have more questions or want to learn more about the CReM, I invite you to check out the website. So last year we started collecting samples to build this bank. And so this cartoon right here is showing the KAT6A protein, and it's identifying the different variants for the samples that we are able to collect. And then we also have the KAT6B protein. Currently, we do not have any samples from last year, but we're hoping to build upon the KAT6A protein samples, but also start adding to the KAT6B protein. So I want to thank everyone who has donated last year, but then also this year as well to building this bank. We do have a blood draw station set up in a different conference room, and there will be a phlebotomist there until noon today collecting blood samples. If you are interested, you can talk to me after the talk as well, and we can show you where that is at. So now I'm going to talk a little bit about what we are going to be doing in the Serrano Lab. I'm currently training at the Center for Regenerative Medicine to learn how to reprogram cells. So the samples that we collect today, I will hopefully be doing the reprogramming for them very soon. And so similarly to what I have stated in all the other diagrams, is it starts with the patients and collecting these samples, and then we're going to reprogram them into iPSCs. And in the Serrano Lab, we're going to do a neural differentiation. And so we're going to differentiate them. They're going to become neural rosettes, which is neural epithelium, and they kind of go in these little circles. And then we're going to go in and we're going to isolate each one of these. And once we have those rosettes, we can go down two different pathways where we can generate brain organoids, which is a 3D structure of these rosettes, or we can have 2D neuronal cultures. But both of these are going to be model systems that we're going to use to learn more about KAT6A and KAT6B. And to give you a little bit of an idea of what this looks like in the lab, Carly, who's currently a PhD student, has been doing neural differentiation for different cell lines that we have in a lab. And this is some of the work that she's been doing. So like I said, we have these neural rosettes, and we go in and we isolate these individual ones, and then we grow them into organoids. So we have this tissue growing on a well plate, and then when we start to see these rosettes, we can set up the microscope here, and then we will actually go in with a glass hook, and we will isolate all of the tissue surrounding the rosette, or if there are multiple rosettes together, we will separate them. And so I have a video to show you what this looks like. So this is the glass hook here, and then this is us isolating the rosette and pushing all of the surrounding tissue apart from it. So once we have that rosette isolated, we can then take it and transfer it into a 96 well plate where it will grow and develop into these brain organoids. This is also some of the work that Carly has been doing in the lab. She's been doing neurodifferentiations and generating these organoids. And so here are some examples for what they look like at day 1, day 7, and day 14. And it's really cool because you can actually look and see them getting bigger as they develop. and we can actually grow them for 3 months and then up to 6 months, which really gives us a lot of opportunity to study the development. And then here's a different picture. So this is what they look like when they're in the dish. The other slide was using a microscope, so they were really zoomed in. So these organoids right here are 2 months old, and then here's a zoomed in picture. And then these ones are 4 months old, and then also a zoomed in picture. So again, you can already start to see the difference from the two time periods and how they are growing and developing. And then this slide is also a assay that Carly did looking at the migration of the cells. So we have the brain organoid right here, and then over here the cells are migrating, and I also can play that video. So this was done just in a well plate, and we were able to see the cells migrating. And then this is my last slide, so I just want to summarize everything that I've talked about and where we want to go in the future. So I started off talking about what is an iPSC and how that's going to help us as researchers, families, and patients learn more about KAT6A and KAT6B. So we're hoping to get as many samples from patients as possible so we can really reflect the patient community. And then once we have those samples, we're going to go through this process of reprogramming and establishing the iPSC lines to use in our own work, but also to share with researchers across the world. And in doing that, we'll have an iPSC bank that can serve as a tool and as a resource for other researchers to test their hypothesis and ask questions to learn more about KAT6A and KAT6B. Specifically in the Serrano Lab, we're going to be using the cell lines to understand cortical neurogenesis in KAT6A and KAT6B using the iPSC neurons. So I just want to thank everyone in the Serrano Lab and at the Center for Regenerative Medicine for supporting and giving me the opportunity to do this work. Thank you to the KAT6 Foundation and also everyone who has been donating samples last year and also this year. If you'd like to learn more about that, you can talk to me. We also have a station set up if that is something you're interested in, and I'm happy to answer any questions.